I will show you today some results that we got in my company, a company that we created now it's 20 years ago. The company is named Plant Advanced Technologies. And this company is organized in different subsidiary companies, or we could say departments. And basically the company, the mother company is doing is trying to discover and biologically assess and industrially produce molecules found in plants or plant extracts more generally. And for this, we are using a greenhouse-based technology. You will see we are growing the plants in aeroponic conditions, not hydro, but aeroponic conditions. And for this, we have two facilities, one in Lorraine, so the region where we created the company, with 3 hectares greenhouse dedicated to this technology. But we have also a settlement in La Réunion Island. It's in the Indian Ocean. It's a French department. And in addition, we have other companies to the group. One is located in Belgium. They are doing biological assessment, targeting dermatology because this is our main market. On the other side, we have a company dealing with production of plant dyes and pigments, because very often the same molecules that we are dealing with in terms of biological efficacy, they are also colored molecules. And we are targeting non-food markets. And we also created two different subsidiary companies. One, Thermesis, is dedicated to developing new anti-inflammation drugs, mainly in dermatology, but not only, anti-inflammation or immunomodulators. And another one, the last we created, is a Silengo. And Silengo is doing metabolic engineering, so synthetic biology, to produce the same molecules that we discover in plants. So that's basically the landscape of the company. But let's go back to some historical points. I'm sure you know these wonderful examples of very important pharmaceuticals, anti-cancer compounds that have been developed, inspired from plants. And this development occurred between the 60s and 80s. And now if you look at the modern days, we can clearly see that plants are now having a declining influence in the pharmaceutical industry. This can be realized, for instance, if you consider that the 10 most successful drugs sold on the market in 2024, 7 out of 10 are biologics, so not small molecules like we could find in plants. And I've done an overview for this today, and I found that in 2024, the Food and Drug Administration, they approved 50 new drugs. And out of these 50 new drugs, just 3 of them were from plant origin. And I would say there are minor medications, not huge medications like anti-cancer compounds. So clearly there's a declining influence. This declining influence started, I think, 2 or 3 decades ago, because the pharmaceutical industry has established new standards of rule, I would say, for the development of new pharmaceuticals. And so they are based on high throughput screening. So they love to screen out hundreds of thousands of molecules each year. And it's been decades that they are developing these molecules on target-oriented strategy. Based on the target-oriented strategy, it means that they start from the protein, the targets they want to affect, and they're trying to shape new molecules based on these targets. Whereas, of course, the plants, when you start with the plant extract, you don't know the molecules that you have inside, and of course, you don't know the proteins, the targets you will address. So that's a major drawback linked to plants. The pharmaceutical industry, to have a drug on the market, you need to have to know the mechanism of action very precisely. And in most cases with plants, when you start with the plant extract, you don't know which target you will engage. Also, you need to have very selective drug. And this is not fitting very well with plant extracts or plant molecules, because plant molecules are prone to give what we call polypharmacology. It means that a single molecule like a flavonoid or the triterpenes that you were talking, Caroline, well, they will address several targets at the same time. And the pharmaceutical industry doesn't like this. And also, last but not least, if you want to raise funds in the pharmaceutical industry for your project, you need to have a patentable drug. And if you're looking at natural compounds, the chemical scaffold in itself, if it's natural, it's not patentable because it was not created by human. So that's another drawback. On the other side, we have plant research giving rise to series of natural molecules. As I said, they are usually poorly selective against human pharmaceutical targets or human proteins, just because they were not shaped by evolution to affect these proteins, but plant proteins or fungi proteins. They are very often moderately efficient. It means that you need a large concentration of your molecule to get the biological effect. Plant extracts, they are difficult to standardize. When you're running HTS, it's better to know the exact concentration of your molecules because it will help defining the activity. When we start with plant extracts, we don't know the concentration. So how can we handle efficient HTS? In addition, plants can be difficult to source geographically speaking, also because 2/3 of the plant biodiversity, terrestrial biodiversity, is located between the tropics. Well, I'm not saying the rest of the world is more stable now, but should be. And the international regulation are restricting access to genetic resources, also for the private companies. That's what we call the Nagoya Convention rules, or access and benefit sharing linked to genetic resources. So what should we do? Should we panic and get rid of plant extracts, or shall we try to carry on the project? Well, that's what we decided, of course, to try to adapt to this pharmaceutical industry strategy. First, I would say that relying on terrestrial plants is still very valid if you're looking to certain types of biological effects. Plants are very relevant to find out anti-inflammatory compounds, just because plants in nature, they have to fight hyperoxidative conditions generated by photosynthesis, generated by UV light, by whatever you want. But plants have very good antioxidant compounds, which turn very often to be also anti-inflammatory compounds. And also, plants were shaped by, in the evolution, with their relations with microbes. And so there's a high chance to find good anti or pro-microbial molecules in plants. Just an example to illustrate this, with 2 research programs we ran for several years, one with BASF, with which we were looking for natural biofungicides found in plants. And the hit rate we had on our plant extract was more than 20%, whereas usually when you're screening out a chemical library, you have 0.1%. This figure comes from BASF itself. So, and again, when we were looking for anti-gram positive and negative molecules, we found that we had more than 10% of our plant extract that were efficient. So, yeah.
Can you give us a little bit more insight about that rate?
Yeah.
What is the criteria of the starting?
Well, they were shaped by evolution, these molecules, to interact with fungi and other bacteria. That's all. I mean, you have more chance to find active molecules in this field than if you run casually from a chemical library. That's it. Okay.
What were the criteria for your starting cohort of molecules?
Well, we select the plants. There are different factors that we use. We can use, depending on the market, we can use ethnopharmacology. Of course, we base on literature when compounds are described to be fungi or antibacterial in this case, or we can go casually also. Okay, but you have a high rate of it when you're looking for antifungal or antibacterial molecules.
If you don't mind, maybe we keep the question for
Sorry.
after,
Yeah,
because
So at PAT, my company, we give attention to most exclusively to plant root extracts. And I'm sure Caroline convinced you why we should pay more attention to root extracts, okay? Because that's where you have more chance to find active molecules because of all of this and arbusculum mycorrhiza I cited, so I'm safe. Okay, I have a short movie to show you what we do in a greenhouse. So I hope it will be readable also with the people which are online. So this is the main greenhouse we have in Lorraine. It's 3 hectares. So we invested more than 10 millions just to give you an insight of the investments in this greenhouse. So we grow different types of plants every year. Usually we screen out 100 new plant species every year, okay? So the plants are grown on tables and just to have access to the roots, and these roots hang freely beneath the table. As you can see, different types of fruits, different colors depending on the plants. And so these roots, these plants are grown in a cultivation zone, and then the plants are moved on the table because it's movable. We carefully select the molecules we want to produce. These roots or plants can be elicited to produce more molecules. It's not so fluid, the film, sorry about that, but so we can enhance the concentration in molecules just by applying plant hormones. So we stimulate, we elicitate, we increase the concentration of molecules, and then at the end, we move the plants to a harvesting machine where basically we soak the roots in a bath of solvents that is adapted to the molecule and the plant we work with. And then after, the plants go back to the growing area. So we alternate growing and harvest, and usually there's one harvest every month. So I think it's the unique factory based on plant aeroponics in the world so far as we knew. Of course, then we have lab control, and we do phytochemical investigation, metabolomic investigations to discover or to follow carefully the molecules of interest. So we rely exclusively on organic agriculture principles, so no pesticides in our greenhouse. As for the print we have in the use of resources, we were surprised to see that we have a very modest use of water in our systems. We use from 40 to 60 millimeters per year, whereas usually with crops, it's more like 600 to 800 in northern part of France. We have a modular system that can be established where you want, now in La Réunion. We have a project in Brazil. And as you see, we are biodiversity friendly. We need more plants every year to screen out the molecules. Just to give you an insight on how we can reconsider the screening of plant extracts, instead of going blindfold, we are using this type of technology that we've developed. And it's also target oriented, just like the pharmaceutical industry likes to do. So we start with a protein here, put in contact with your plant extract. Only one or a few molecules will bind to your protein. You can get rid of the unbound compounds here. You denaturate the protein and you get a chromatogram where basically you have fished out only the molecules that are stick to your target. And that's an example here. That's the full chromatogram with all the composition of your extract. And you can see that, for instance, this pig here, this molecule has been selectively enriched as compared to this chromatogram in the chromatogram here below, it shows that this molecule is probably the best inhibitor found in a plant extract. So in a matter of few hours, we can sort out which molecule is affecting directly your target. And in this case, we fit with the requirements of the pharmaceutical industry in terms of discovery. So now I would like to illustrate how we can address the One Health story with our technologies. Everybody speaks of One Health, but is it real? Is it real? I mean, can we really develop a single molecule for different markets, human, animal, plant health? And for this, I will give you some elements on this molecule. This is one of the molecules we prefer in my company. It's 3,5-D-caffeoylquinic acid. So it's a secondary metabolite or specialized metabolite, which is quite spread out in plant kingdom. You can find it in a lot of plants, but in larger quantities in sweet potato, artichoke, and green coffee beans. And this molecule, it's not us, has been recognized as extremely active in terms of anti-inflammatory process. And more than this, there are numerous papers describing its neuroprotective effects. And when we got in contact with the doctors working on this, they told us we cannot develop it because it's not available on the markets. I mean, if you want to buy this molecule, it will cost you at least 500 euros a gram, and it's too expensive to be developed into something like a pharmaceutical or nutraceutical compound. So what we did is that we tried to understand how it's produced in plants, and we developed a microorganism able to produce this molecule. So we deciphered the biosynthesis, and then after, we pushed the molecule in the nutraceutical market with the natural scaffold and with some synthetic derivatives into the pharmaceutical market. So how the synthesis works in plants? Well, 3,5, the molecule is there. 3,5-diCQ is produced from caffeoylquinic acid, so the monocaffeoyl ester. And so far, before we were interested in this, the main road was supposed to be an HTT enzyme using a caffeoyl-CoA ester to shape this molecule. But it's not very active because we worked on this enzyme. It's not very efficient. And we thought that in plants like sweet potato, which can produce larger amount of this molecule, up to 5% of the dry weight of the roots, so that's quite a lot, there must be another enzyme which is more active. And that's what happened indeed. We found out an enzyme belonging to the GDSL family, which is a lipase esterase family of enzymes. And it's much more active to produce 3,5-DCQ. You can see here the scale is much important here. So after we discovered this enzyme and characterized it, we introduced the coding gene into Pichia. We developed the Pichia strain, which is able to produce the enzyme at a high level. And now we have an industrial process because it works at 500 liters now scale. And we have a process where basically feeding the enzyme with chlorogenic acid, so the substrate, we can form 3,5-DCQ. And we have a project with a Canadian company working in the food sector, where basically we will transform green coffee extract, that's the black chromatogram here, into enriched extract in 3,5-DCQ, thanks to the use of this enzyme. So as you can see, when we operate the enzyme during 24 hours, we low down the concentration in 5-CQA, which is a substrate, and form the product. Then the same molecule was used to develop some drug candidates. And for this, we were inspired by nature. In nature, diversification of secondary metabolism is a way that used by plants to produce more and more chemical entities. And one of this diversification is showed here. This is prenylation, where basically it can work on an aromatic structure. You add a chain of carbon, and this change very much the activity of the starting molecule into something more lipophilic, with more cell membrane affinities, so more hydrophobic interactions with proteins. You increase the bioavailability, you increase the Rho scavenging property, and these molecules are much more active in lowering down inflammatory process and cell proliferation than the non-prenylated ones. So what we did was, starting from 3,5-DCQ, we produced through chemical synthesis some carbamide derivatives of different chain length. And it happens that the C8 turned to be extremely active in as an anti-inflammatory molecule. Is here shown on mice. So this mouse is inflamed every day, and from day 7 to day 10, we apply the molecule in a patch in the skin, and as you can see, after a few days, the mouse is perfectly clean. So it's very efficient. We found out the mechanism of action of this molecule. It affects the synthesis of this protein, I-kappa B zeta. And as far as we know, it's the only molecule described so far, or patented so far, to target this protein. And so we are compliant to the mechanism of action that must be well known. And the same 3,5-DCQ today is also being tested by us in collaboration with INRAE in Avignon and a private company against green aphid, Myzus persicae, because this molecule was described to be somehow active in some papers. And we have designed new molecules inspired from this natural scaffold to get more activity. So yes, the same molecule can be used to tackle inflammation, memory loss in case of 3,5-DCQ in green coffee, and natural insecticide. In terms of perspective, I think we can reconsider plant chemistry in many ways to fit the requirements of the pharmaceutical industry. So far, what we do when we work on the discovery of molecules is finding the right key to unlock this locker. But in the pharmaceutical industry, it's been more than a decade that they are using another approach, which is called fragment-based drug discovery. Instead of starting from perfectly finished and big molecules, you start with spare parts, smaller chemical entities, and you assess how these smaller parts can fit with a part of the lock. And this way you can design with retrochemistry a new compound which is perfectly adapted to the locker. And this, of course, can be used, and project we have is to use fragment-based drug discovery, but based on libraries of natural fragments. Because today, the pharmaceutical industry is using chemical libraries, which are quite different than the natural fragments that you can find in plants. This is small fragments. This is fragments of less than 200 grams per mole. And you just need, I would say, 200 fragments to cover a huge chemical space. And this approach can be potentialized, of course, by artificial intelligence using molecular networks, QSAR, and graph theory. So that's something I think that gives many promises for the future. Then I have another question for you. In a post-genomic and post-artificial intelligence era, will we still need to go botanical trekking to discover new molecules? Because if you rely on chemical libraries being natural, is it still useful to go in nature? Well, the answer is yes, of course. That's some pictures I made recently in a botanical trekking in the Vosges Mountains, where we were picking up mosses, bryophytes, to make some investigation on these wonderful plants. So not only it's inspiring, it's beautiful, but of course we know that we have very intricate interactions between plants, microbes, insects, and this is a chemical diversity that you will never get into chemical libraries. Then I had it just as an example of how plants can inspire us. Since Stephanie was speaking in her introduction, she said I would talk about molecular farming. It was not my intention, but we worked on that for many years. And during, it was a Friday afternoon, one of my collaborator, he came to me and said, you have carnivorous plants for your students. Shouldn't we use them to produce recombinant proteins? That's a crazy idea because, well, we know that carnivorous plants, they excrete large quantity of proteins in these droplets. This is the Drosera plant, or in the picture of Nepenthes plants. Can it be used to produce recombinant proteins? Well, the answer is yes. And we developed a transformation protocol either for Drosera or Nepenthes. And we did even more than that. We found out the first virus infecting Nepenthes that we used as a transgenic vector. This was done with a famous virologist in Bordeaux. Sorry.
Congrats.
Oui. Voilà. Merci. And we were able to produce human interferon and human intrinsic factor. This is a gastric protein. And so it works well. But we were facing some resistance in the pharmaceutical industry when we proposed to use this type of plants, because the pharmaceutical industry, as any industry, they love to have standards and keep their standards on the tracks. So it's difficult to make them change. And we decided in 2017 to stop this project. And for those of you interested in metabolic engineering and in molecular farming, if you follow what happened in Medicago in Quebec, the company which was most advanced in producing proteins from plants, they shut down the company 18 months ago. I want to thank all people at PAT who contributed to this work. Not only PAT, people in different research centers. For instance, the story of 3,5-TCQ in plants was deciphered with Université de Lille and Université de Lorraine. And thank you very much.